Okay, welcome to AP Lab 5 that we did a while back in our cellular respiration unit. If you remember the joys of glycolysis and the Krebs cycle and the electron transport chain and all of that put together being the process of cellular respiration. You might also remember we did a lab to investigate how different conditions can affect the rate of cellular respiration in a living thing. And in this lab, our living thing was peas, which are shown in this container. And in order to measure respiration, we put some peas in another container. The container has two openings. Into one opening, we placed a probe that can measure the levels of oxygen in this enclosed chamber, and the other probe that we inserted into the chamber is a CO2 probe that can measure the amount of carbon dioxide in the enclosed chamber. Okay, and by measuring those two values over time, we could get a sense of the rate of cellular respiration in peas under differing conditions. So remember, in a lab situation where you're doing an experiment, we always want to be able to tell what our independent and dependent variables are. In this lab, we were testing a couple of things. We were testing the effect of germination on cell respiration rate and the effect of varying temperatures on cell respiration rate. Germinating simply means sprouting, growing, and what we see here are some dry pea seeds. These are seeds that would be dormant that you might buy in a seed envelope at the garden center to plant. And they are not germinating, they are not growing in any way, they are just sitting there waiting to be planted. These peas, however, are sprouting. As you can see, their little roots are coming out. They are, in fact, germinating. They are starting to grow, which means they are carrying out cellular activities. Okay? Now, we compared these germinating seeds to non-germinating seeds, and we also compared germinating seeds at regular old room temperature to germinating seeds that had been chilled down to ice cold temperatures. Okay, so those are our independent variables: germination and temperature. That's what we purposefully affected. Okay, now what did we measure as a result of what we did? We chilled them down, we germinated them, we didn't, whatever. What we measured as a result of those activities was how much oxygen the peas consumed and how much CO2 they produced. Now, both of these are measures of cell respiration that's occurring. Why are they measures of cell respiration? Because if you will recall, the cell respiration equation is that glucose plus oxygen gives us water, CO2, and energy, of course, in the form of ATP. Now, that means in order to sprout, these little peas are going to be using up glucose that's stored in their own bodies in the endosperm of their cells, the cotyledons, and they are going to be using up O2 from the atmosphere in this chamber. Okay? They also, as a result, will be giving off CO2. So what we would expect then, as a result of an experiment in which, let's say, we're just looking at germinating peas, standard room temperature, what would we expect them to be doing in terms of what we're measuring? We're measuring O2 and CO2 levels in this chamber. We would expect that the oxygen levels would go down because we are using up the oxygen in this enclosed chamber, and we would expect CO2 levels to go up because we are the peas should be giving off CO2 if they're doing cell respiration. Please note it's really important that this chamber is an enclosed space, otherwise O2 and CO2 could be coming in and out, so it's very, very important that it's an enclosed space. Now, generalized concepts for this lab. First of all, you really do need to understand cell respiration. You do need to also remember that this is not something that just happens in animals. It also happens in plants. It's just that plants can also do photosynthesis to make their own food, but they must break it down using cellular respiration. Now, you need to know about the experimental design aspects of it. What's our control treatment? Well, whatever the norm is. So let's say that you could argue it's germinating peas at room temperature. Some people might argue that it's the dormant ones. They shouldn't be doing anything. You can make different arguments for different treatments here. You do need to know some constants, but things should be the same for each experiment that you run. You should, the chamber itself, the size of the chamber, the number of peas perhaps that you put in the chamber, anything that can be held the same from one treatment to the next should be. And you might benefit you to consider ways that we could easily alter this experiment to measure different things. We could use little animals instead of peas. We could do different things to the peas. We could expose them to different kinds of conditions. Maybe I don't know, light and dark, or any number of different conditions that would we could just test to see if it had an effect on their respiration rate. Okay. Now the results we've already discussed. We would probably expect. Well, actually, we discussed the levels of CO2 and O2, but what would we expect temperature and germination to do to those levels of O2 and CO2? In terms of just sheer respiration, is respiration occurring or not? If we decrease the temperature, we might expect that the respiration rate would go down because, of course, if you'll remember, if we decrease temperature, we are lowering the kinetic energy and therefore reducing the number of molecular collisions that are going to occur, and so respiration would slow down. I think in our peas it didn't quite turn out that way, but this is typical of what one would expect for most chemical reactions. As you decrease the temperature, you decrease the activity. But germination, we would expect, since the peas are being active and they're growing, we would expect that respiration rate to go up. So in other words, more CO2 produced and more O2 consumed, so O2 levels would decrease. This is a graph that you might make given the data that we collected with this. Remember, in this graph, we are looking at the milliliters of oxygen consumed over time and minutes. We have nice scales on either side, and what we see are 4 lines because we are looking at the germinating. This is corn, but same difference. Germinating at room temperature, germinating cold. Non-germinating at room temperature, non-germinating cold. So whatever you measure, you plot it, draw your lines. Also, again, don't forget about a rate calculation. If we want to know what is the rate of respiration, y2 minus y1 over x2 minus x1. If we wanted to just take this data point and our 0 point, you could do that. In that case, for example, if we do that for the germinating at room temperature, then it would be, our x would be 20 minus 0. It looks like our 20 reading is at 1.6 mLs minus 0. So whatever 1.6 divided by 20 is, that would be our rate. And you would want to go ahead and calculate that out and use your units. What are the units? That's milliliters per minute. Okay, don't forget your units. All right, here's a sample essay in which this lab was the focus. So they're giving you data that was collected over time, and it's asking you to graph it, calculate the rate of oxygen consumption, account for the differences between the different experimental treatments, and then explain the experimental apparatus you would use. That's this lab directly. Okay, so that's that, and I hope that's helpful to you, and I'll catch you at the next one, Lab 6 coming up. Thanks for watching.